Assalamualaikum students. So this is Irfan sir here. We are discussing enzymology. We have discussed enzyme composition, factors affecting enzyme activity, and also enzyme inhibition is there. And also we discussed activity of enzymes through two hypotheses. One is Koshland's induced effect model, and also we have discussed that is lock and key model. And most importantly, we have discussed enzyme activation energy is reduced, is decreased by the enzymes by which the rate of reaction is increased. Now, this is the last topic of the enzymes, that is enzyme classification. Now, before discussing the classification of enzymes, let us see a little bit about the enzyme nomenclature. Earlier, when enzymes were isolated or discovered, they were given arbitrary names like pepsin, trypsin, chymotrypsin, renin, and these names have been kept as such, and we use these names even today. Later on, it was agreed to add suffix ase to the substrate. Later on, it was agreed to add suffix ase to what? To the substrate. For example, there is a substrate lactose. Lact was basically kept there and ase was used as a suffix. So the enzyme got its name lactase. Similarly, there is sucrose. Sucrose is again a substrate, right? And enzyme, it is sucrase. Similarly, there is maltase, there is dextrinase, right? Now, as far as classification of enzymes are there, according to International Union of Biochemistry and Molecular Biology, there are six major classes of enzymes. There are six major classes of enzymes. Class number one is oxidoreductases. This is the class one, that is oxidoreductases. This is the class which basically catalyzes redox reactions. You know very well that is a reduction and oxidation. There is a gain of electron and oxidation is a loss of electron. In other words, you say there is a removal of hydrogen, transfer of hydrogen, and addition of oxygen. So in short, they catalyze the redox reactions, reduction as well as oxidation. For example, there is an enzyme lactate dehydrogenase. There is an example of enzyme succinate dehydrogenases. So all dehydrogenases, they belong to which enzyme class? Oxidoreductase, that is class 1. It's a very simple topic. Class second is called transferases. As the name itself speaks, that is, it is going to catalyze the transfer of groups. Of course, other than hydrogen, right? Hydrogen transfer is not included in this class because it is included where in the class 1, that is oxidoreductases. So transferases, what they do, they catalyze the transfer of groups. Like there is an example. This is the first reaction of the glycolysis. Hexose, there we will see the glucose. Glucose is hexose, is a 6 carbon sugar. So it's a hexose and there is an ATP, right? So one phosphate is transferred from ATP and is given to what? Hexose. So it becomes hexose 6 phosphate. So the phosphate is transferred. That is the reason the enzyme is called hexokinase, and this hexokinase, it belongs to what? It belongs to the class second, that is transferases. And remember one thing also, whenever you see the name kinase, kinase means those enzymes is a class of enzymes which do the phosphorylation. Phosphorylation means they add phosphate to the substrate. Always remember, whenever you read hexokinase, whenever you see hexokinase or any other kinase, right? Kinase always refers to the enzyme which is doing the phosphorylation, that is addition of phosphate. The third class of enzyme is called hydrolase. Hydrolase. It is very simple. Hydrolases are a class of enzymes which break the bonds in the presence of water, with the help of water, right? So it catalyzes hydrolysis. Lysis means break the bonds with the help of water, right? For example, ester bond, ether bond, peptide bond is there, glycosidic bond is there, right? All these bonds, they are present in the biomolecules, you will come to know. And example of this class is digestive enzymes, and there is an acetylcholine esterases. So simply saying, hydrolases are the class of enzymes which break the bonds, which catalyze hydrolysis, break the bonds in the presence of, yes, water. Like ester, ether, there is peptide and there is glycosidic bonds, and these bonds are present in the biomolecules, right? And for example, there are digestive enzymes. You know the digestive enzymes, whether they are working in intestines, right? Small intestine, they require water, right? So they are the hydrolases. And there is one more example. There is acetylcholine esterase. So this is the third class of enzyme. Now let us see the fourth class of enzyme. Fourth class of enzyme is lyases. This is the fourth class of enzyme that's called as lyases. Lyases basically, they also break the bonds, they remove the groups, but not the way the hydrolysis are doing. Means other than hydrolysis, they don't require any water, right? So they don't break with the help of water. They do the removal of groups, they break the bonds, but the mechanisms other than hydrolysis. Example is there. We have a compound here, fructose 6 bisphosphate. It is also an product of this. It's present as an intermediate in the glycolysis as well. So it is fructose bisphosphate. It is acted upon by aldolase, and aldolase breaks it. It's a 6 carbon compound. It's a 6 carbon compound. It breaks it into 3 carbon 2 compounds, each 3 carbon. Glycerolide 3 phosphate, and there is dihydroxyacetone phosphate, right? So it's a 3 carbon compound. It's a 3 carbon compound. So it breaks the bond here and give rise to the 2 compounds, 3 carbon each. So this is an example of the lyases. Remember that it does not require water. It's going to break the bonds, but the mechanisms other than hydrolysis. Now the fourth class of enzyme is called isomerases. Now isomerases, you know the isomerism. You people know the isomerism from the very beginning. Isomerases, what they do the interconversion of isomers, whether they are optical isomers, geometric isomers, positional isomers. They do the conversion. One form of isomer is converted into another form of isomer due to the molecular rearrangements, right? For example, there is an example. This is glycerolide 3 phosphate. It is isomerized into dihydroxyacetone phosphate, and the enzyme is isomerase and is called triose phosphate isomerase. So it's very simple to understand. Just now the last class of enzyme, it is a class 6 and is called as ligases. Ligase is basically a Latin word liger, which means to bind, to link. So ligase is a class of enzyme. Ligase is a class of enzyme which condenses two substrates together. Which combines, which condenses two substrates together, and the reaction is driven by the energy derived from what? Derived from the ATP, right? So this is driven by what? ATP, it requires energy. Example, acetyl-CoA is there, acetyl coenzyme A, plus there is carbon dioxide, and the energy is provided by the ATP, and the enzyme is here, acetyl-CoA, and means that acetyl coenzyme A carboxylase. And the product formula is malonyl coenzyme A plus ADP, and what is there? There is inorganic phosphate back, and the energy release is utilized by linking the carbon dioxide, condensing the carbon dioxide and acetyl coenzyme A. So these are the 6 classes of enzymes, right? Now these 6 classes of enzymes, they have been given a sequence, like class 1 is oxidoreductase, right? But how you are going to remember this? There is OTH, OT, and lil. Some students remember it is OT hill. They do remember this as OT hill, HILL. But I suggest you just remember this acronym OT lil, because if you remember OT hill, oxidoreductase, transferase, there is what? Hydrolase, isomerase, lyase, and ligase. In this acronym, the sequence is not correct, but here it is correct. What's the class 1? Class 1 is oxidoreductase, right? So this is the class 1 oxidoreductase. What's the second class? Second class is transferases, right? What's the third class? Third class is hydrolases. Now what's the fourth class? Fourth class is lyases. If you see here, first class is oxidoreductase, second is transferase. What is the third? Hydrolase. Now what is the fourth class? It is lyase, but lyase is here. You understand? So if you want to remember it, you can just remember OTLIL, OTHLIL, little. OTLIL, you just remember this acronym OTLIL. And if somebody is comfortable, OTHIL, you can do it as well. But in the sequence, it is OTLIL. So oxidoreductase class 1, transferase class 2, hydrolase class 3, and lyase is a class 4. And of course, this is the fifth class, that is isomerases, and the last class, class 6, is the ligases. So here the sequence is perfect. OT, LIL, L-I-L. I hope it's clear. So this is all about the classification of enzymes and the 6 classes. So it's very easy. It's an easy topic. Now there are some terms regarding the enzymes. I will discuss these terms also. Now there are some terms regarding the enzymes. The first one I discuss here is zymogen. Zymogen. Zymogen is simply a term given to inactive enzyme. When the enzyme is inactive, it is called a zymogen. For example, there is prorenin, it is converted into renin. For example, there is pepsinogen, it is converted into pepsin. So pepsinogen is inactive enzyme and inactive enzyme is called zymogen. Number one. Number two is enzyme. Enzyme basically means catalytic antibody. Catalytic antibody, right? So when antibody is acting as an enzyme, when antibody has a power of catalysis, that antibody is called as enzyme. The third term is isoenzymes or isozymes. Isoenzymes or isozymes, they are the multiple forms of the same enzyme. Multiple forms of the same enzyme catalyzing the same reaction. Multiple forms of the same enzyme catalyzing the same reaction may be present in different tissues. I will quote an example here. So isoenzymes or isozymes, what they are? They are the multiple forms, distinct forms of the same enzyme and catalyze the same reaction. For example, there is an enzyme called LDH, lactate dehydrogenase. It has 5 isozymes. Right? Simply we write them as LDH1, right? LDH2, LDH3, LDH4, and there is LDH5. Got it? Now this LDH1, LDH2, LDH3, LDH4, and there is LDH5. LDH1 and LDH2, it is present in heart and RBC. In the heart and the red blood cell, you will find LDH1 and you will find LDH2. And LDH3, you will find it in brain and kidney. In brain and kidney, you will find LDH3. Now LDH4 and LDH5, it is present in liver and skeletal muscle. Right? So liver and skeletal muscle. So I repeat, isozymes, different forms of the same enzyme catalyzing the same reaction. Right? Now example is on here LDH. LDH has 5 isozymes, 5 isoenzymes. LDH1, 2, 3, 4, and 5. LDH1 and 2 are present in heart and RBC. LDH3 is present in brain and kidney, and LDH4 and 5 they are present in liver and skeletal muscle. Just a tip, you must understand this also. LDH1 and LDH2, since it is present in the heart muscle cells, they also act as cardiac markers there. Cardiac markers, right? They act as a cardiac markers. You can check them during there is heart attack, during there is some destruction of the myocardial muscle cells. Although not too much specific, but we can use them. Cardiac markers, LDH1 and LDH2. They when the heart muscle cell dies, these markers come out and they just accumulate in the blood, and their levels in the blood tells a lot about the disease, tells a lot about the extent of the disease in the heart or destruction or the death of the myocardium in the heart. So their levels are checked in the blood. I hope it's clear. Now listen students, this is all about the enzymology. We have completed this chapter also, and now we are going to take the cell cycle or cell division we will take, right? And one thing important, each year in NEET as well as in AIMS, you will find always 2 to 3 questions asked. Even in the previous year, even in AIMS or even in a NEET previous year, you will find 2 to 3 questions asked in AIMS as well as in the NEET. So be very, very careful. Do everything what I have discussed so far in the enzymes. Build your concepts, especially when I discussed about the factors on which enzyme activity depends, Vmax, and there is Km. What are the different, I draw some graphs here, relationship between the substrate and the enzyme, relationship between substrate and the temperature and the curves and all those details. So you will be asked, questions are regularly asked on the enzymology, and 2 to 3 is the trend. So it's very important for the NEET aspirants, those who are preparing for the NEET. Now see you in the next lecture, see you in the next video with cell cycle or cell division. Till then, Allah Hafiz.